Introduction. Welcome to the fascinating world of nucleic acids. In this course, we will explore the intricacies of nucleic acids, their structure, function, and importance in various biological processes. Nucleic acids, namely DNA and RNA, are fundamental molecules that play a pivotal role in the storage and transmission of genetic information. Now, let's delve into our first topic, Introduction to nucleic acids. Nucleic acids are large biomolecules found in all living organisms. They are composed of building blocks called nucleotides, which consist of a sugar molecule, ribose or deoxyribose, a phosphate group, and a nitrogenous base. The two major types of nucleic acids are deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. DNA serves as the genetic material in most organisms, carrying the instructions necessary for the development, growth, and functioning of living systems. RNA has diverse functional roles, including acting as intermediaries in protein synthesis, catalyzing biochemical reactions, and regulating gene expression. DNA is a double-stranded helical molecule that forms a ladder-like structure known as the double helix. The backbone of DNA is composed of alternating sugar and phosphate groups, with the nitrogenous bases projecting inward. The four nitrogenous bases found in DNA are adenine, A, thymine, T, cytosine, C, and guanine, G. The sequence of these bases determines the genetic code, which contains the instructions for the synthesis of proteins. DNA replication occurs during the cell division process, ensuring the faithful transmission of genetic information from one generation to the next. RNA, on the other hand, differs from DNA in both structure and function. It is typically single-stranded and can fold upon itself to form various secondary structures. RNA also has ribose as its sugar component and uses uracil, U, instead of thymine, T, as one of the nitrogenous bases. There are several types of RNA with distinct functions, including messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, RNA. mRNA carries the genetic information from the DNA to the ribosomes during protein synthesis, while tRNA brings the amino acids to the ribosomes for protein assembly. RNA, along with various proteins, forms the structure of ribosomes, which are responsible for protein synthesis. The genetic code is a set of rules that determines the correspondence between a sequence of nucleotides in mRNA and the specific amino acids they encode. It is a universal code shared by all organisms, from bacteria to humans. Protein synthesis, also known as translation, occurs in the ribosomes and involves the conversion of the genetic information carried by mRNA into a sequence of amino acids. This process is essential for the synthesis of proteins, which play crucial roles in cellular structure, function, and regulation. While DNA and RNA are the most well-known nucleic acids, there are also non-canonical nucleic acids with unique structures and functions. These include synthetic nucleic acids, modified nucleotides, and nucleic acids with unusual base pairs. Non-canonical nucleic acids have applications in areas such as nanotechnology, molecular diagnostics, and gene therapy. Understanding these alternative nucleic acids expands our knowledge of their potential for biomedical research and the development of novel therapeutics. To study and manipulate nucleic acids, scientists employ various techniques. Some of the basic techniques include DNA extraction, polymerase chain reaction, PCR, gel electrophoresis, and DNA sequencing. DNA extraction isolates DNA from cells or tissues for further analysis, while PCR amplifies specific DNA sequences. Gel electrophoresis separates DNA fragments based on their size, allowing for visual analysis. DNA sequencing reveals the precise order of nucleotides in a DNA molecule, providing invaluable information for genetic research. DNA replication is a highly precise process that ensures the accurate duplication of genetic information. It involves the unwinding of the DNA double helix, followed by the synthesis of complementary strands using each original strand as a template. The end result is two identical DNA molecules. DNA repair mechanisms constantly monitor and correct errors that arise during DNA replication or due to environmental factors, such as radiation or chemical exposure. Failure in DNA repair can lead to genetic mutations and an increased risk of diseases, including cancer. Gene expression is the process by which genetic information is used to synthesize functional gene products, such as proteins or non-coding RNAs. Cells tightly regulate gene expression to ensure proper development, growth, and response to environmental cues. Regulation can occur at multiple levels, including transcriptional control, post-transcriptional modifications, translational control, and post-translational modifications. Dysregulation of gene expression can lead to various disorders, emphasizing the importance of studying gene expression regulation mechanisms. DNA recombination is a natural process that rearranges genetic material between different DNA molecules. This process plays a vital role in the generation of genetic diversity, evolution, and the immune response. Genetic variability is the foundation for adaptation and evolution. It allows organisms to respond to changing environments, withstand diverse challenges, and maintain genetic robustness. Understanding the mechanisms behind DNA recombination and genetic variability provides insights into the complexity of life. Telomeres are specialized structures at the ends of chromosomes that protect them from degradation and fusion with neighboring chromosomes. They consist of repetitive DNA sequences and associated proteins. Telomerase is an enzyme that adds repetitive DNA sequences to the ends of chromosomes, counteracting the natural shortening of telomeres with each cell division. Dysfunction of telomeres and telomerase is implicated in aging, cancer, and various genetic disorders. RNA interference, RNA, is a powerful mechanism that regulates gene expression by silencing specific genes or reducing the amount of their corresponding RNA molecules. This process occurs through the action of small RNA molecules, such as microRNAs, miRNAs, and small interfering RNAs, siRNAs. RNA has emerged as a valuable tool for gene manipulation and therapeutic interventions. It has the potential to treat genetic diseases, viral infections, and even certain types of cancer. The study of nucleic acids has unraveled the underlying mechanisms of various diseases. Genetic mutations in nucleic acids can lead to inherited disorders, while alterations in gene expression contribute to the development and progression of complex diseases, including cancer, cardiovascular diseases, and neurological disorders. Understanding the involvement of nucleic acids in diseases provides opportunities for the development of targeted therapies, diagnostic tools, and personalized medicine approaches. In conclusion, nucleic acids are the fundamental molecules of life that drive the inheritance of genetic information and regulate biological processes. Through the exploration of topics such as DNA structure, RNA function, gene expression, genetic variability, and disease mechanisms, we gain a deeper appreciation for the intricate workings of nucleic acids. As future medical professionals, a solid understanding of nucleic acids and their implications in health and disease will enable you to contribute significantly to the advancements in research, diagnostics, and therapeutics. Embrace this journey of exploring the remarkable world of nucleic acids and their role in shaping life as we know it. Best of luck in your studies.